DNA replication. We know that DNA is self-replicating molecule. All living creatures have capacity to reproduce because of this characteristic of DNA. DNA is semi-conservative. In 1958, Matthew Meselson and Franklin Stahl performed experiment to prove that the DNA is semi-conservative by using heavy isotope of nitrogen, that is N15. They grew E. coli in medium containing ammonium chloride, in which nitrogen is heavy isotope, for many generations. As a result, DNA get incorporated into newly synthesis DNA. This heavy DNA can be distinguished from normal DNA by centrifugation in a cesium chloride on basis of density. Now they transfer E. coli, of which DNA is incorporated with N15, into a medium of normal ammonium chloride, that is N14. After a definite time of interval, DNA extracted from the culture and centrifuge, the E. coli from first generation had a hybrid or intermediate density. Again, hybrid DNA allowed to grow on normal culture, and after some time, it again tested. Second generation shows a light N14 state, as well as hybrid state. This proves that DNA is semi-conservative, that is, one complete strand of daughter DNA is derived from parent DNA. Mechanism of DNA replication. In living cell, DNA replication takes place under the action of certain enzymes, of which DNA-dependent DNA polymerase is important enzyme. This enzyme is important because both strand of DNA is used as template and polymerize the chain of deoxynucleotide. And we know, one nucleotide contain a deoxyribose sugar, a phosphate group, and a nitrogen base. That is purine and pyrimidine. Nitrogen base form a bond with the first carbon of pentose sugar, that is, deoxyribose sugar, to from nucleoside. And phosphate group get attached to fifth position carbon of pentose sugar to from nucleotide. Two nucleotides get attached by forming a bond with third carbon of pentose sugar with phosphate group. And finally, two strands of DNA get attached by hydrogen bond. For simplicity, let's take another structure of DNA. We know DNA is composed of two polynucleotide chains. Both chains are anti-parallel and complementary to each other. It means if one chain has polarity 5:3, the other has 3:5. And replication of DNA always take place from 5:3.